Jasmine, it's yours.
All right. Hi everybody. Thanks for staying on. We're really excited to next have Yamuna Krishnan tell us about her living history. So in the interest of keeping things moving and in time, I'll pass it on to Yamuna.
Okay, thank you so much for that. Like many others on this series, my path in science is also very unusual. I've worked in three different continents doing fundamental research in three completely different cultural contexts. And I've also experienced places where there was no element of research at all. And this gives me a very different view on how we practice science. And this kind of like unusual trajectory could certainly not be planned. And even though at every stage I had a crystal clear idea of what my next step was going to be, a different choice arose, another exciting but more risky alternative. And I always ended up rejecting the path of security and going for the one where I felt like I'm probably biting off more than I can chew. And I'll talk about each one of these pens. So my father is an architect and my mother is a literary figure. And so I grew up in Chennai, a place in south of India, surrounded by books and T-squares and blueprints. And actually, neither of my parents took an active interest in my schoolwork. They both said, we have our jobs. You are expected to do your job, which is your homework. And when there's going to be no policing, you get in trouble, it's your problem. You're on your own. But they got me a microscope and a little lab equipped with a few harmless salts that didn't do much, but it was enough to sort of kindle in me a love for experimentation. And I was never the class topper. I loved all the extracurricular activities in school, but I really enjoyed actually the lab practicals, lab work. And it often made me wonder, whether sticking kids' noses into textbooks is the best way for them to learn science. Because I, for one, learned it best by actually trying to do experiments. And I always thought I'd end up being an architect like my father because I had an eye for buildings and spaces and drawing. But my final exam in math was a total disaster. And I wound up taking chemistry because one of my older cousins was studying chemistry and he had hand-me-down textbooks. I could get that for free, so chemistry it was. And so I went to this small liberal arts college in Chennai, and there were two fabulous teachers in chemistry, and thanks to them I fell in love with chemistry. And they sort of understood this and they sort of fed me advanced level study material, and I just lapped it all up, right? And meanwhile, my elder cousin who finished his undergraduate, he went on to study masters in chemistry at the best place in Chennai, which is called IIT Madras. And I thought, well, this must also be my logical next step. So that was what I was going to do. And I wrote only 3 entrance exams, just completely clueless. But I got offers from all 3. And the Indian Institute of Science admitted me directly into a PhD program. And I chose that mainly because I was very enamored by the beauty of the main building. And because the questions I was asked in that interview was the toughest compared to all of the others. So I joined a PhD program without knowing what PhD meant. I'd had no idea of what research was. I just thought it was like, reading books and writing tough exam questions. That's very ignorant. And so I chose my PhD lab based only on the science. But the lab environment that I experienced was quite toxic. And to survive, I developed a very strong network of friends that spanned an array of departments in the Indian Institute of Science, as well as a lot of other labs. And this was very formative for me because it put me on a path of interdisciplinary science because I learned to communicate science with everybody and kind of understand what they were doing as well. And I met and married a physicist along the way. And during my PhD, I learned about how to control the self-assembly of lipids by adjusting molecular scale interactions and to use these lipids for materials or biological applications. And during my final year, I was exploring the application of a lipid I'd made in bringing about DNA transfection, and this was in a collaboration with a professor in the biology department called Professor Sandhya Vishweshwaraya. And as a graduate student, I was quite inspired by her dedication to science, her scholarliness, She's always above, all the petty internal politics, never let it get to her. She never jockeyed for awards, never indulged in this quid pro quo that we have. And she is still and had she was the single biggest influence on my thinking and on my value system. And so while working in her lab, I realized that, I wanted to explore the chemistry of lipids, but connected to biology somehow. But all the leaders in the field were in the US, right? So that was where I was going to go and do my postdoc. And then in my final year, my husband decided to go to Cambridge for a postdoc. And so it was expected that that is where I was going to go too. So I had to change my research area in order to follow him. But I got a couple of offers in Cambridge. One was from an older, highly accomplished and well-known fellow of the Royal Society. The other was this young chap, very sharp. But I chose to work with him because he had good ideas and I knew he would have time for me. And that contact time with him would make me a better scientist. And so I joined him. Shankar's lab environment was the polar opposite of my PhD experience. And that taught me a lot because to this day, the most important thing in my lab is first the environment and then the science. And I applied my knowledge of self-assembly in Shankar's lab to sort of study how DNA self-assembled into this odd four-stranded structure called a quadruplex. And the one thing that I remember at that time was that I was generally hungry for knowledge about all areas of chemistry, not just what I was working on, because I just loved going to department seminars and sort of peppering the speaker with questions. And Shankar noticed all of this and encouraged me to apply for various fellowships and awards, and some of which I was shocked that I got. Then I decided, I wanted to bring back more DNA chemistry into India because I saw this gap in our country. And so I started applying for faculty positions, thinking that the most appropriate place would be a chemistry department, either in a place like TIFR Bombay or Indian Institute of Science in Bangalore. And so I spent quite some time targeting these places because that's where I was going to go. And then, then I met Vijay Raghavan and Jeetu in Cambridge. And I had such a fantastic time talking to them that I visited NCBS too during my tour of different institutes in India. And the day at NCBS that I spent just changed everything. Those days it wasn't as famous as it is now, but the science and my future colleagues was so fantastic that I couldn't picture myself anywhere else. And I loved it so much that I signed the first offer that they made. Big mistake. I learned always to negotiate after that, even if I really wanted something. Meanwhile, my marriage broke up, but that was good because it improved my mental outlook. And then the first 2 years at NCBS, I realized that I knew no biology at all. And I struggled to sort of withstand this harsh light under which biologists sort of viewed my kind of science because it's chemistry, right? And that's because chemistry is an inventor's science and everybody else was engaged in the process of discovery. So the mindset itself is different. And my field was structural DNA nanotechnology. And the rest of the field was engaged in sort of folding DNA to create stunning shapes on the nanoscale. And in contrast, I was trying to see if I could make simple assemblies, but ones that had function in living systems. And because there was a very strong school of endocytosis in terms of Jeetu's lab, who was studying endocytosis, I tried to see if I could use my nano devices to probe the pH of endocytic organelles. And so over 7 years, I laid the foundation of this concept where we showed that nano devices made out of DNA could sense pH in a range of organelles that were present in live cells and live worms. And then I realized that if I had to expand the potential of this platform to its full form, I would need to do chemical synthesis and I would need to have access to samples from human patients. And I realized that that was not going to happen here at NCBS because the institutional priorities were different. And I was okay with that. But the day I realized this, I went and I told Vijay that, I would need to look elsewhere. And Vijay, ever the optimist said, great, just see what's out there and generate a couple of offers, Yamuna. And this was liberating for me because it allowed me to fully and openly explore all the possibilities out there. And it was during one of the talks that I gave during this, my search for my next step, that I encountered Professor Chuan He, who recently got the Wolf Prize. And he suggested, I should visit his place at U Chicago, even though I already had a soft offer from my alma mater, Cambridge. And though I always knew that I was going to go to Cambridge, I visited U Chicago anyway, only because of Chuan. And again, the energy and quality of science here just completely reoriented my plans. And I moved to U Chicago because I knew that I could do chemistry at U Chicago, but also the medical school was virtually next door, just separated by a few corridors. And so I could see enormous possibilities for growth at U Chicago. But what I didn't realize was that, of course, I found out later, I had no prior track record in the US. And so it was basically a complete and total reset to zero for me. And it took me about 7 years to get my first NIH grant because the system here in the US is favored to, it's sort of weighted in favor of 4 human phenotypes that I don't have. But my department was really fantastic. It is actually a fantastic department because they prioritize only quality science. And I was never made to feel any the lesser for not bringing in federal funding. So I then started a company based on our technology. And over the next 7 years, we sort of fully developed this sensing platform. And then the unpredictable happened. Through a series of impossible events, I ended up meeting my life partner, who happens to be a Nobel laureate. His name is Jack Szostak. And then the pandemic struck. And so one day, we're all working from home, right? So he was watching me sort of rushing around doing 3 things at the same time while working from home. And then he just told me, very calmly, just slow down, think more deeply and do what matters. And this changed my approach completely. I guess I was also probably ready to hear that for it to sink in. And I learned to access a greater depth of quality thinking. And in doing so, I realized, I had built every tool that was needed if one wanted to build a quantitative electrochemical model of organelle membranes. And then suddenly, all our grants got funded at the same time. You see, the only biological membrane for which we have an electrochemical model is the plasma membrane that was developed for neurons by Hodgkin and Huxley in 1952. And so this is now what I'm excited to do as we move forwards, which is to build an electrochemical model for all kinds of other biological membranes that are present in organelles. And so that's where I am. And to reach here, it takes a village. For me, it takes a town. And I'm just grateful to all the people along the way who I have tried to highlight, who have helped my journey. And so thank you. Happy to take any questions.
Thanks so much, Yamuna. I think we're running a bit behind, so perhaps we won't take too many questions, but feel free to throw all the appreciation and everything in the chat. I just started at UChicago this January, so I'm really happy to meet you as a colleague now, and hope we get to meet in person soon.
Thanks, Yamuna. Closing the recording.